So now I am going to discuss on how Taylor performed. Scientist Taylor in 1957 performed his experiment of identifying the model of replication of the chromosome. Okay, how the replication of chromosome happened. He took the legume called the broad bean, Vicia faba. In that Vicia faba, he made some improvisations within his experiment in order to understand that how the DNA, how the chromosome was multiplying. Okay, how the chromosome was multiplying. He performed on the chromosome. Okay, he was unaware. What Taylor did, he was unaware that the replication was happening solely in the aspect of or with the functionality of DNA. He concluded that the chromosome was replicating. The DNA, he was not aware that the DNA was actually replicating and giving rise to the two bodies, two parts, the two arms of the chromosome, the two chromatids, the sister and the non-sister chromatids, the entire body of the chromosome. He was unaware of the fact that the DNA was actually responsible for for doing the replication and again transforming itself or coiling up itself in the form of the chromosome, and further the chromosome was showing that characteristic feature which he concluded at the end of the experiment. So what he did, Taylor, if I put the heading as semi-conservative replication by Sir Taylor in 1957. Okay. Semi-conservative to 19. Now, let me draw the structure of an X chromosome. Okay, not a normal chromosome, whatever the structure is. He considered this. Why? Okay, let me tell you one thing in this aspect. Always remember, in most of the experiments which are depicting any chromosome in any bioscience book or anywhere, okay, or over the internet also, people always draw the structure of an X chromosome. Why? Because in the autosomes that are present within the human body, or in the autosomes that are present across the body of the other species, there are the different kind of autosomes are there. Okay, different bodies, different organisms have different designation of the sex chromosome or the allosome. In human beings we have X and Y, and in case of the other species, lower groups of animals like bees and all, we have ZW, ZZ, XX, XY in case of Drosophila and all. Then ZM, ZZ. These are ZO, ZW. These are different kind of organizations of the sex chromosomes that are present across the diversity of the species. In case of birds, in case of reptiles, the representation of the sex chromosome is different. The depiction is different. In case of human beings, it is taken as X, and this X type of chromosome is also found in the body of the other organisms, some other organisms, like in case of the Drosophila melanogaster also we can find XXXY is there. In human beings already it is present. In case of other some plant species are there, very few unique plant species are there where the chromosomes are being depicted with X and X only, okay, in the form of X. Why this X chromosome is being considered in the role play of any kind of explanation? Because X chromosome is having a complete organization of non-sister as well as sister chromatids. So in order to show the process of division of the chromosome or the karyokinesis, that is nuclear division, the X chromosome stands to be the symmetrical chromosome by which the clear depiction of the division of the chromosome can be depicted. The symmetry of the chromosome is very well organized. So if I draw the normal chromosome, Suppose this be the normal chromosome of the beans of broad beans of the Vicia faba. Okay. What Taylor did, Sir Taylor did, he took a plant species known as broad bean, which was a legume. Okay, legume basically. Scientific name is Vicia faba. Broad bean. Scientific name is Vicia faba. Okay. And in 1957, what he did in order to understand the model of replication, that how the replication was actually happening, how did one chromosome give rise to another subsequent chromosome, he performed this experiment. What he did, he grew the broad bean cells, Vicia faba cells, in a medium of tritiated thymidine. Okay, he grew this in the medium of the tritiated thymidine. Tritiated thymidine is actually a type of isotope, a type of element which is radioactive in nature. Okay, now this is the normal. This one is normal chromosome. This one is normal chromosome. Now what he did, after a subsequent period, he grew this Vicia faba broad bean cells. He cultured them within a medium of tritiated thymidine. So he put these chromosomes in the culture medium containing tritiated thymidine, which is a tritiated thymidine, which is a radioactive element. Okay. Now this cells of the Vicia faba broad beans, when they were being grown for a subsequent period of time in this culture medium, which was having the tritiated thymidine radioactive element, he has seen or observed that after subsequent generations, after few generations, this chromosome Vicia faba converted into a radioactive form. Okay, converted into a radioactive form. This is the radioactive form of the chromosome. This is actually the radio labeled chromosome. Okay. Now, what he did, he isolated this radio-labeled chromosome and replaced this radioactive medium, with a non-radioactive medium. Okay. After a few generations, when he found or observed that the chromosome now become radioactive, how they became radioactive? This chromosome incorporated this tritiated thymidine element. This tritiated thymidine element got incorporated within the chromatids of the chromosome. These are the chromatids. These two are known as the sister chromatids because they are present on the same side of symmetry or same side of the centromere, and as well as these two are the sister chromatids, and opposite chromatids, these two are non-sister chromatids. Okay. Now, when tritiated thymidine got incorporated within the chromosome, the chromosome got radioactive. Now he replaced this tritiated thymidine medium with a non-radioactive medium. Okay. in the non-radioactive medium what happened? He subjected this same chromosome. He subjected this same chromosome in a non-radioactive medium which was not having trishaded thymidine. This was radioactive and this is non-radioactive. Non-radioactive. Now in this non-radioactive medium, this chromosome or the cells of the Vicia faba beans also grew. This is actually culture medium or a type of growth medium which was having, the previous one was having the trishaded thymidine and this was not having trishaded thymidine. Okay. Now in this case, this particular cells grew, the Vicia faba beans grew, and he found that after the first generation, of the growth, the culture medium was showing intermediate radioactivity. Intermediate or hybrid radioactivity, because the radioactivity got less. It was reduced to half. Why? Because this, in the non-radioactive medium, when this radioactive chromosome multiplied, they formed one radioactive section of the chromosome. Okay. And one non-radioactive section of the chromosome. So this was not non-radioactive, but this was radioactive. Okay, and the chromosome multiplied into two cells, right? Two chromosomes. So again, another such chromosome was formed. Another similar chromosome was formed, which was having a radioactive section along with a non-radioactive section. So this black colored area is the non-radioactive zone. Okay, and again this part here is the radioactive section. Radioactive. So he found that this, when he isolated the chromosomes from this culture medium, okay, he found that the radioactivity of the chromosomes was reduced to half in the first generation. Okay, after first generation means one cycle division, one division of the chromosome. He found that the radioactivity of the chromosomes were reduced to half. Why? Because one arm of the chromosome was having entire radioactivity and another arm or another half was not having that. Or again, another chromosome, one chromosome is forming two chromosomes. In another chromosome also, one arm was showing the radioactivity and the other part of the chromatids or the other arms was not showing the radioactivity. Okay. Now, there was equal concentration. These two were presenting the equal concentration. And now in the second generation, that is after the first generation was over, in the second generation he found that when he isolated the chromosomes again, this one is the first generation, let me write first generation. Now again in the second generation, second generation, what will happen? This particular chromosome will divide into 2 chromosomes and this particular chromosome will divide into 2 chromosomes. Now in the second generation, again the same way of replication was conserved. This chromosome is forming these 2 chromosomes particularly. Okay. And here also the same thing happened. This chromosome was forming these 2 chromosomes. These 2 chromosomes. Now, in this case what happened, in the second generation also he have seen that in the first generation there was an equal concentration of the radioactivity, that is the entire radioactive chromosome, both terms were radioactive, it was highly showing radioactivity, and in the first generation the radioactivity was reduced to half. Okay. Both the chromosomes or all the chromosomes were showing equal amount of radioactivity. Okay, here I have seen. And in the second generations, there was a difference. In the second generation, there was a difference. Half of the chromosome was not showing radioactivity, and half of the population of the chromosome was showing radioactivity. These 2 particular chromosomes were showing non-radioactivity, non-radioactive, whereas these 2 chromosomes were showing radioactivity. Here also this was showing radioactivity. So 50% of the chromosome population was not radioactive and the other 50% or other half was radioactive. Why? He concluded, Taylor concluded that this is the parent chromosome, right? Okay. Now in this parent chromosome, this particular half of the chromosome will replicate or give rise to a fresh new strand or replicate in a new chromosome arm, and this parent arm of the parent chromosome will also give rise to a particular new chromosome arm. Now in the byproduct or in the 2 new different daughter cells that have been formed from this particular parent cell or mother cell, what will happen? Along with this parent radioactive chromosome, there will be a non-radioactive arm of the chromosome. This is the non-radioactive arm, and along with this non-radioactive arm of the chromosome, there would be another non-radioactive arm of the chromosome in another daughter cell. Why this is happening? There is a common dilemma, confusion in this part, that when it is semi-conservative means he named it as semi-conservative mode of replication. Semi-conservative means one parent arm of the chromosome was brought forward or carried forward in each of the daughter cell. That is, if this parent arm is carried to this daughter cell, now the daughter cell will also have another arm radioactive, because this radioactive chromosome arm is replicating, means it is producing an identical copy of itself. So that radioactive parent arm will also produce a radioactive arm, a new radioactive arm of the chromosome. So this black colored arm of the chromosome would not be black, it would be red instead. But this is not happening in this case. This is not the obvious case. Why? Because the medium here, that particular medium here was being changed to a non-radioactive medium, right? Now when the non-radioactive medium is being reacting with this chromosome, the chromosome will try to incorporate non-radioactivity within itself. Just the way the chromosome incorporated radioactivity in the first case, in the second case the chromosome will try to incorporate non-radioactivity within itself. So in that case what will happen? The parent arm, though it is radioactive, the fresh new daughter arm that would be produced, that would be non-radioactive. And this parent arm, which is non-radioactive, that will obviously produce another daughter arm which is radioactive. And in this case also the same thing is happening. For this particular chromosome also the same thing is happening. So 50%, that is half, two of the chromosomes here, these two chromosomes, this one and this one, are showing non-radioactivity, and this chromosome was showing radioactivity, but lesser, or the radioactive intensity was reduced to half, considered to the intensity that was present in this medium or for this particular chromosome, right? So this is what, that is how I concluded that the semi-conservative mode of the replication is nothing but one parent arm will be present with the daughter arm, and a freshly produced arm of the chromosome will also be present within the daughter chromosome. Okay. So in the course of formation of a new cell, one parent arm is going along with the formation of a daughter arm. That is how the parent chromosome is dividing itself to produce two new subsequent daughter cells. So this is what Taylor concluded via his experiment of semi-conservative model of replication. Okay.